Well, it doesn't get shared with the classroom. All right, we're getting started. A lot of people here tonight. Yep. Yeah. Always. Okay, so we're gonna do cell cycle 2. It's more focused on uh S phase, G2, and M phase. Um, so me and Gabe, the duo. All right, so quick overview cell cycle. Start G1. That's the gap or growth phase, up to whatever you want to call it. So the cell is making sure that it has enough nutrients, enough organelles, cell machinery in order to divide. And so we're also ensuring no mutations in the DNA. And get to the restriction point, and the cell then commits to the cell cycle, or it goes into apoptosis if it thinks it won't be able to make it through. And so, yeah. And in the case that the cell feels that it is not ready just yet to replicate and not divide, before the restriction point, it can go into G0, and it's a non-dividing state. And so we move along to the part that we're focused on this week. So S phase, G2, M phase. So the S phase synthesis, go on to DNA replication, and there is the end of phase S phase checkpoint. And so then you move along to G2. It is the second growth phase, stage, whatever you want to call it. And the cell is preparing for chromosome segregation. And there is the very important G2M checkpoint. If you are not too clear on it just yet, I'd recommend going back to the lecture videos, spending a little bit of time on the G2M checkpoint because it is fairly important. And then you move along to M phase or mitosis, whatever you want to call it. And here we're talking about chromosome segregation up until cytokinesis. So there's PMAT, prophase, metaphase, anaphase, telophase, and then cytokinesis. And that is the route that the cell follows in order to finish dividing. So moving along to S phase. So entering S phase, we are going to talk about pre-IC formation. And so this is a little bit of a review from the end of last week and, what Laura went over in lecture this week, at least so far. And so you need a pre-RC to be assembled and ready in order for the pre-IC formation to occur. So the pre-RC, pre-IC is the middle of the replication bubble of where replication should start. And if you look at figures about how that works, you'll see that, at least in like in vitro figures, you'll see that they kind of look like vacuoles in like a medium if you look at it. And there can be multiple replication bubbles along the DNA. As you probably can think about it, there's no way you could just replicate from one stage and have it ready quite enough. And so there are multiple firing points that happen. And so this complex requires active CDK for formation. And I'm not going to go 100% over the complexes that you need to know. The drawing is a little bit complicated. But you do need to know that the MCM complex, also known as helicase, is the center of it. And so after pre-IC forms, the polymerase can be recruited. And once the polymerase is added, then the complex is known as a replosome. And like I mentioned beforehand, there could be multiple replisomes along the DNA. And so the origin of replication is where the firing is going to start. And that is basically when helicase unzips DNA and replication starts at either end. And so that's when you enter S phase. And although there are multiple origins, not all of them fire at the same time. Some earlier, some later. It depends on multiple variety of different factors. So these are the steps. So SCDKs inhibit new pre-RCs from forming. And so you only want to replicate the DNA once per cycle. What does that mean? Well, once the replication starts, you don't want it to like continuously stop, start, stop, start. And so S-cyclin CDKs phosphorylate ORC to prevent binding of CDC6. So this is on a this is before the pre-RC is formed. And once CDC6 is phosphorylated, it goes on to degradation through ubiquitination and or ubiquitin mediated proteolysis. And so once that happens, the MCM cannot be recruited and you don't have the formation of the pre-RC. And once MCM2 or MCM gets phosphorylated as an MCM27 complex, then it gets exported from the nucleus. And I would write that down and I would know that MCM gets exported once it's inactive, because that was a question I had during one of my exams when I took the class. So it might come back, it might not come back, but I would keep that in mind. And so the cohesin rings form around the replication or the replicant DNA. And so that's what keeps the chromatids together. And so it prevents the chromosome segregation until the cell is 100% ready. And so when after all the steps required for cytokinesis to occur, that's when cohesin gets cleaved. And so very important, that C step right there. Cohesin rings must be present in S phase in order to function in M phase. And we will see why. And so there's an end of S phase checkpoint, and the cell is checking for DNA damage. It is checking to see if its DNA is completely replicated. P53 activation can happen in this phase as well, and we saw P53 was a tumor suppressor earlier on. Very important molecule. Okay, who's brave enough for a question? What's P53 do? Oh, you have a question. What's your question? Huh? Yeah. What's P53 do? Yep. What does that do? So ultimately, what does it do? There you go. That's what I wanted. What was your question though? In this phase. Yeah. It won't happen unless it's does it happen? Do you know if it happens in G1, Gabe? Because I know it can sense for DNA damage, but I think that's mostly on the DNA replication basis. Yeah. Yeah.
Yeah, I think it's not.
G1. It can happen in G1.
Yeah, I saw it on like the previous exam, only focused on G1. G1.
Yeah.
It was, you know, I do vaguely remember that they go over like top two.
Yeah. So to answer your question, yes. Yeah, so it constantly checks for it, but P53 is not the only molecule that checks for DNA damage. And so in this checkpoint, replication can be stopped temporarily. And as we saw in the previous unit, if it halts for too long, then it just goes into apoptosis. All right, Gabe, that's you.
All right. TA Genius Gabe here with another episode of 285. Okay, so in G2, the cell goes back to growing again. It's done with its DNA replication. So now it needs to make sure that the DNA that it replicated was replicated correctly and checks for DNA damage. And it's not going to go into M phase unless there's some like mutation we'll talk about. It's not going to go into M phase until the cells are fully grown and all the DNA has been checked. And this phase is important in order to get the cell to the appropriate size manner before dividing to make sure that there's no mistakes in the DNA and all that. Make sure there's like enough resources and organelles made so that each independent daughter cell can have enough to survive. And M phase CDK needs to be activated in order to go into M phase. We'll go over that in more detail. So at the beginning of G2, MCDK levels are rising, but are inactive, and it takes CAK, phosphorylating MCDK, with an activating kinase in order to activate it. However, there's also going to be an inhibitory phosphate put on by WEE1, and this inhibitory phosphate is going to be stronger than the activating phosphate. And so when MCDK has both of these phosphates, is going to be inactive. And yeah, so then later on in G2 phase, after the cell is determined that it is large enough to go into M phase, CDC25, which is an activating phosphatase, is going to be activated, and it takes off the inhibiting phosphate from MCDK that was put on by WEE1. And then once that's taken off, the MCDK is now active and pushes the cell into, with high enough levels of active MCDK, the cell will go into M phase. However, this process accelerates very quickly. Once a few of MCDKs are activated, it accelerates very quickly the level of, you'll see on graphs, the level of like MCDK activity, because of the fact that there's this positive feedback loop where once you activate a few of these, active MCDK acts as an activator for inactive CDC25 phosphatases, which in turn help activate the activate more of the inactive MCDK by removing more of the phosphates that are inhibiting it. And this cycle keeps going on and on until the levels reach high enough levels to. Oh, and the other thing that it does is that it inactivates WEE1 from the previous slide. So it's no longer going to be adding these inhibitory phosphates. And that's going to push at high enough levels, it'll push it into the cell into M phase. And so for M phase, just a quick overview of like mitosis in general. In prophase, they're going to have active MCDK just initially starting to activate some other enzymes that are going to cause the breakdown of the nuclear envelope. So the nucleus is going to have its membrane breaking down. Chromosomes will start to condense into like the typical shape that we think of when we think of chromosomes. Then pro-metaphase, the microtubules will be growing and shrinking, and they're going to get aligned and start attaching to the microtubules over here. And then metaphase, the chromosomes are going to start to align along the metaphase plate here. Chromosomes are going to have their proper spindle attachments at the kinetochores, which is the center of the chromosomes. And then, what? Oh, my bad.
Centromere is the center
Oh my God.
that the kinetochore gets built upon.
Oh, my bad.
That's the center of the chromosome.
Okay, don't listen to me.
Listen to Gabe, it's not.
Okay, Yeah, so anyways, these we're gonna have like a push and pull mechanism we'll talk more about here to sense that there's equal tension on both sides. In metaphase, they're basically gonna get lined up. Spindle assembly checkpoint's gonna be here, which is gonna be a big thing to remember for this class in general. Which is why it's bold on here. And then in order to like pass on from metaphase onto anaphase, it has to pass, it still has to pass the symbol assembly checkpoint, which is like the biggest checkpoint since the G2 one we just saw. Once it passes that, anaphase will occur where they get the chromosomes or the sister chromatids get separated to each side to each daughter cell, and then telophase and cytokinesis is the separating of these cells. The nuclear envelope starts to reform and chromosomes decondense, and basically it's the opposite of prophase, like what we saw. Rather than sister chromatids, yeah. Yeah. It's the same thing.
Yeah, same thing.
Yeah, she just doesn't like the term. So yeah, so a little more detail. So prophase, in prophase, what happens is that it started off because of the active MCDKs, and so the active MCDKs are going to activate or phosphorylate certain proteins that are going to cause ultimately these effects that start off prophase. So they're going to phosphorylate lamins, which are going to be proteins that break down the nuclear lamina. They're going to also phosphorylate condensin, which is going to start to condense the chromosomes from looser, unpacked DNA to more tightly packed structures that we see here, and various other maps and motors in order to get the mitotic spindle assembly going. And so for sequential phosphorylation, the idea is that phosphorylation needs to happen in a certain order. And the big idea here is just that like the CDKs are master cell cycle regulators. So they're not going to be phosphorylating every individual protein in the cell that like causes the effects of that happen to in order to like start off like mitosis, for example. They're going to like prime down or like activate like downstream other like downstream activators in order to, and then those individual activators will have like a downstream effect that will ultimately cause the effects that need to be done. Yeah. And so spindle assembly is going to occur, it's going to start to occur in prophase, but this is like a picture of metaphase. And so this is going to be, there's like different sections of the cell with different sections of microtubules that are involved. So there's three sections. There's three types of microtubules. So the first type we'll talk about is aster microtubules, and they're the ones who are going to be connected to the cell cortex, which is basically like the cell membrane out here. And then they're going to be connected to the spindle poles, and they're going to help keep tension, pulling apart these chromosomes. And then kinetochore microtubules are ones that are connected to the kinetochores on the kinetochores of the chromosomes. And then
again, chromosomes don't have kinetochores. Kinetochore is a protein that binds to the centromere of the chromosome. And that's what the kinetochore microtubules bind to pull the chromosome apart.
So this, I want to review what you just said, so basically formatting what they said attach the chromosome without color. Chromatin? Well, wait, chromatin?
Kinetic core.
Wait, and then, kinetic core is a protein. It's good. There's a slide on it.
Yeah, we'll go more in
There's a
detail.
slide on
It's okay. We'll just. No, you're good. There's a lot of terms. It gets easier when I go. I would hold off on the question until we get to the slide because there's more details there. Just will show you the differences. So yeah, the kinetic core is going to be connected to the microtubules here, the kinetic core microtubules, which are also connected to the spindle poles. And then lastly, we have these interpolar microtubules, which are also attached to the spindle poles, but they're attached to each other through these motor proteins that we'll go over as well. And they grow from the spindle poles and they're going to be overlapping in the middle. And again, I'll go over more detail about the proteins there. So the kinetochore is a large multi-protein complex, and it's built on the centromere area region of each homologous chromosome or sister chromatid. And there's a bunch of proteins involved with that, but we'll go over in more detail as well about that and how it's involved with the spindle assembly checkpoint. But part of its job is to attach the spindle microtubules to some of these proteins within the kinetochore itself. So there's a push and pull mechanism that helps drive parts of this, each part of the sorry, I'm just drawing a blank here on the spindle assembly. And so the spindle itself. And so the overlapping ones, the overlapping microtubules, the interpolar ones, are going to have different motor proteins on them that push apart. And so the main idea overall, though, is that the whole spindle formation requires these motor proteins of kinesins, which walk towards the plus end, and dyneins, which walk towards the minus end of microtubules. And they're going to walk towards opposite ends of microtubules to create opposing pulling forces that stabilize the spindle. And that's for these ones right here. And then an example of a motor protein that's interpolar is the plus end directed EG5, which EG5, which is like, this is like a zoom in version of this. And so it's going to have proteins on, it's going to have dimers on each side that are both walking towards the plus end of each microtubule. And each microtubule is anti-parallel like DNA. And so because of that, they're going to be pushing apart from each other as they're walking towards the plus end of each one.
This was an exam question. So EG5 is a tetramer. EG5 is kinesin 5. And so what that means is, just to add to what Gabe was saying, it has 2 pairs of motor domains at each end. So it also has 2 pairs of tails at each end. And so that's how it can walk on both microtubules. Because as we talked about in the microtubule unit, for motor proteins to walk, they need to hydrolyze GTP in the tubulin dimers. And so that's how they move. And the fact, like Gabe was saying, that there are 2 different plus ends and 2 different minus ends allow these microtubules to be pushed apart by these motor proteins. Like the figure is showing on the bottom right, there's a plus sign. And on the top left, there's a plus sign. The only kinesins that move towards the minus end are the kinesin 14s. And so because EG5 is kinesin 5 and it has tetramer shape, it is able to move 2 microtubules along with respect to each other. And that is how So if you think about it, from like a technical standpoint, the two minus ends are being pulled together because the two plus ends are being pushed apart. And so they're walking in opposite directions, right? So. I think it's best for them. No, go back.
You wanted to show the arrows
no.
or no?
So. Explain to the phones. But right to the
Wait, they
recording.
can they not
They
hear?
can't hear me, but I'm doing this for them if they're here.
Oh, okay, okay.
Talk over. So. This motor protein, this motor head, or these two motor heads, are walking towards the plus end of this microtubule, while these two heads of the same kinesin are moving towards the plus end of this microtubule, which causes them to push the opposite directions and push the microtubules away from each other. So basically, the way you want to think about this is the minus ends of the microtubule are being pulled together, while the plus ends of the microtubules are being pushed apart. So these two are going the same direction. The minus ends are going the same direction, while the plus ends are going to opposite directions. And slide. And so that's what this means right here. So the minus end of this microtubule is being pulled in this direction, and then the plus end of this microtubule is going that direction. And so what you want to say is, so you guys remember what? Actually, I should probably talk this right here. So if you remember well about the MTOCs, they are minus ended, right? You remember the MTOCs? Microtubule organizing centers? Yeah. So they're minus ended, which is what is shown right here. And so this plus end of this microtubule is going this direction. Well, this plus end of this microtubule is going the opposite direction. What happens then is because they are going to opposite directions, the two microtubules are being moved apart. So in the labeling, there's egg 5, it looks like a dumbbell, like the one you lift. Like Gabe pulls like 35s on each arm. He's huge. So dyneins, which is the fish looking one, right? They go towards the minus end. I mean, there's a specific family of dyneins that walks towards the plus end that I don't remember right now. But as far as we're concerned for this class, and unless she doesn't specify it in lecture, dyneins go to the minus end. Okay. And so the dynein is what's going to help this microtubule go towards the minus end. Right? And the kinesin 5, or the egg 5 in this case, is what's going to help push these microtubules apart from each other. What does this do in the ultimate spectrum of cytokinesis, or M phase really? It helps the cell sense enough pressure in the microtubules so that the chromatids can get pulled apart. So ultimately, what's going to happen is this microtubule is going to be pulled towards the MTOP. And that's going to cause this left chromatid to be pulled apart from its other sister chromatid. It's like kind of confusing if you don't see it in like a video. But basically what the cell is doing is it's ensuring that these two can be pulled apart. Yep.
Right, is there a video link on the?
There should be a video on the folder.
Oh,
The video folder is like honestly like one of the best resources Laura has available. So the egg 5 is walking towards the plus end of either microtubule, right? And so the MTOC is the minus end of the microtubule overall. And that figure that we had in the last slide, so this figure right here, this figure right here, you can't really tell because it just looks like the minus end ends right there. The minus end doesn't end right there, or at least in the specific case of the chromatid pulling mechanism, it doesn't. Oh, okay, so it doesn't end like literally like an inch away from each other. It's like a microtubules are like very long. And so egg 5 is walking towards the plus end, right? And so it pushes these spindle poles apart. It's pushing these two apart. So basically you have imagine there's like egg 5 is on my arm. It's doing this. The microtubules are overlapping, and the ultimate end goal is for them to do this.
Yeah, and you're asking the dynein. So dyning's gonna be on it's a myosin-directed protein, and it's going to be found. It's going to be found on these microtubules. For the recording, it's going to be found on these aster microtubules, as well, like on the cortex right here, cell cortex, like on the membrane, and then they're also going to be found on the microtubules themselves. Does that make sense? This one? These ones, yeah, egg 5. Left, this one right here. This is also a dyne, but yeah, but we just like, what's that? Yeah.
This one, I just want to ask like
Oh,
which
yeah.
microtubules it pulls, like pulls that one or this one?
It pulls this one, but the thing is, honestly, for our class, like, we just focused on these, like this right here, and this right here.
So
So like overlapping, the ones connected to the microtubule, the ones connected to like the cell membrane. Like, yeah. Does that make sense? Are there any more questions? We should probably check the chat.
No, there's nobody in chat.
Oh, there's nobody in. Oh, really?
It's just a recording uploaded.
Oh, okay. All right.
No,
go
over the aster dynes.
Astrodynes? Okay.
So there's different dynes. So there's a dyne for the interpolar microtubules, which is the one. Yeah, so there's interpolar dynes, and so they pull the spindle poles together. And so they walk towards the minus end of overlapping microtubules, so the interpolar microtubules, right? And so they hold on to the overlapping. So they're kind of doing what egg 5 is doing, besides the fact that they're not touching it. They're not touching it, right? Okay. And so the aster microtubule dynes, which are the ones that are far away. Laura's talking about how they're closer to the membrane of the cell. They're not in the interpolar region of the cell or of the spindle. And so they pull the spindle pulls apart. So basically what that means is they're increasing the distance between the spindle poles. And so one end of the dyning is attached to the cell inner membrane, the inner part of the cell membrane, if you understand what I mean.
I was gonna say just 80 and 19.
Sorry?
No.
It's just it's lodged, it's not lodged onto the cell wall or membrane, it is attached to it. And the other head of it, or the other side slash end of the dynein, is what's gonna be attached to the or walking on the microtubule. And so, ready? So the spindle poles at the edge of the cell are being pulled apart with respect for one another. So if you think about it in a geometrically, like in a geometric way, if you have so, get my wallet out. Oh, so the closer together, so they're all attached to the MTOC, right? So pretend the MTOC here. The closer together that the spindle poles are, the closer to the center of the cell that the MTOC is going to be. And so the farther apart that they get, so you see how this, pretend there's like a circle right here. The farther apart that these two ends get from each other, the farther from the middle of the cell that the middle is going to be. And so what that basically does is they're going opposite directions, and this is getting farther into the edge of the cell. You understand? I can. The dyneins, the white dyneins on this graph, they go apart. And as far as the MTOCs, so the MTOC is right here. And they're pulling the microtubule. They're moving the microtubule. And so they are pushing these apart in a sense that if they were closer here, this would be farther into the middle of the cell. And then the farther they get from each other, the closer out, or I mean, the farther out this is going to be. Can you?
So it's the end of the cell. This is the MTOC. This is the interpolar microtubule. This is the chromatid. Right? And so the active microtubules are right here. The dyneins are going to be right here. Okay? Done. So basically what you want is that the MTOC gets closer to the edge of the cell, pulling the chromatid farther out. And then what results of that is dynein go farther along the edge of the cell. Yeah. So what happens is this, because of this decreases as they get farther along. You picture that?
So the microtubules, they will be each other. Like, they can, really go back instead of being.
Well, they can get degraded, and that's also a way of making sure that it's all part of that. But the dynein is walking on essentially what's happening.
Okay. So to continue along, okay.
Okay, so so we're at metaphase right now, and so the way that it's going to be there. So what's going on at metaphase is that we have cohesin rings, and they're holding together the sister chromatids or homologous chromosomes, and they're still held together at the kinetochore, the sister chromatids, and there's equal and opposing pulling forces that cause them to line up at the metaphase plate in the cell. And so in order to go to anaphase from metaphase, the cells have to pass what's called the spindle assembly checkpoint. And it just ensures that all the sister chromatids are aligned and the spindles are attached properly at the kinetochore. And they want to make sure this happens so that non-disjunction doesn't occur, which is basically just making sure that, one of the daughter cells doesn't get like an extra chromosome pulled towards it. You want to make sure that there's an even spread of chromosomes to each daughter cell. And so what happens is that what starts off this mechanism is that Mad and Bub are going to sense their proteins in the kinetochore. They're going to sense microtubule tension on the kinetochore, and they're going to sense if there's equal tension on both sides from each microtubule on each sister chromatid. If there is equal tension, CDC20 is then going to be released by Mad and Bub. So Mad and Bub are holding CDC20. They're going to let go once it's time to go into anaphase, which is sensed by equal tension. It's released. CDC's 20 release is going to cause, it's going to bind to APC, and it's going to activate APC. And APC is a protein that is a ubiquitin ligase, so it's going to degrade. It's going to tag proteins to get degraded. And so CDC20 is going to activate APC. It stays bound to it. In its active state, this APC and CDC20 complex is going to target this other set of proteins. It's going to target specifically securin, and it's going to tag securin for degradation. And this is important because securin is holding on to this enzyme called separase, which is capable of cutting the cohesin that's holding the sister chromatids apart. It's the only thing preventing it from the cell from going into anaphase. So, okay. And so once this securin, it's almost like holding hostage the separase, and once the securin, the kidnapper, you could say, um, gets taken care of by the APC, tagging it for degradation, this separase is now free to go and do its job of cleaving the cohesin, which is holding the sister chromatids, homologous chromosomes together. And they're finally able to separate because there's that pulling tension that was already there. And with this pulling tension now, the chromosomes get pulled apart to each side in order to form two different daughter cells. And this is during anaphase. So this whole mechanism I just talked about right now is spindle assembly checkpoint. Again, it starts from MADBUB sensing the tension, letting go of CDC. CDC activates APC, which then tags securin for degradation, which lets go of separase. Separase, when active, is going to cut the cohesin, which is going to basically start anaphase off. So anaphase, there's two parts to anaphase. There's anaphase A, which is the part focusing on the chromosomes being pulled poleward. So it's basically just the mechanism I was talking about with the cohesin. Like after it's degraded, the chromosomes can be walked by minus directed motors, and so as the motors walk toward, they have a part attached to the kinetic core, but they also have a part attached to the microtubule, and as they start walking towards the minus end over here, it's also going to lead to the destabilization, degradate, and the depolymerization of the microtubule at the plus end due to like dynamic instability. And the kinetic cores, so the kinetic core microtubules are going to shorten, and the spindle poles are going to stay in place. Anaphase B focuses on the poles being pushed and pulled apart. So for anaphase B, it's focusing on the spindles lengthening and pushing these poles apart. And then it's also focusing on the forces pulling the poles apart on the ends over here by the aster microtubules. And as well as like the pushing and pulling. So part of that is the growing of the plus ends of the interpolar microtubules. And so as these egg 5 start walking towards the plus ends, these microtubules start growing, and this starts to get longer so that the cell can grow to a size where it can be cleaved in two to create two different cells. And then finally, telophase and cytokinesis is the last step in M phase, where sister chromatids have now reached the opposite poles, and a cleavage for cytokinesis. Cleavage for is created by a contractile ring shown right here, made of actin myosin. It creates an indentation in the cell membrane, as you can see here, that deepens until it hits the spindles that's left between the two nuclei, and then eventually split. And then there's also for part of this is going to be for like telophase. Part of this process is going to be due to the fact that M cyclin CDK activity is dropping, and so the spindle is going to disassemble, and the chromosomes are starting to revert to their state of being chromatin that's loose. Goes from sausage to spaghetti, and the nuclear envelope is going to form around the DNA, which is basically the recreation of the nucleus and nucleolus. yeah, so the main ideas of the cell cycle are that there's 4 different steps, G1, S, G2, and M, which can be further subdivided up into the different stages we talked about today. The cells,
What's the state of the phase? I know what are the last thing in the last.
set of pieces, yeah. Nice. Nice. You'll ace the next exam. Cell cycle, it involves the cyclical activation and deactivation of proteins, a lot of times specifically with CDK cyclins. And for all of these, CDK for each stage has to be bound to cyclin, and it has to be phosphorylated in order to be active. And sometimes it has to be also not deactivated by another phosphate, like in G2 with WEE1. And CDK has regulates various proteins in different phases of the cell cycle, depending on what it needs to do. So different phases of the cell cycle have their own cyclins, and each time it needs to go into a new phase, the cyclins are created and degraded for each part of the respective cycle that they're helping with. And the regulation for proper functioning requires a deactivation and activation and inhibition of all these proteins. Oh, it's supposed to have both of our names on it, but thanks for coming. Do you guys have any questions? I didn't even make these. I cannot say, I plagiarized it. Later, I have to split. I hate when you are gone. I'll miss you. ... Periodically. Just kidding, guys. I'll miss you all when I graduate.
Yeah, I can graduate with this.
Thanks for coming.
We appreciate your presence.
Wait, how do I end the recording? How do I stop the recording?
All right.
Oh wait, I'm